Hello everybody, this is Peter Cooper. Welcome to today's NCBI Minute webinar on the SRA Run Selector. The answers and questions that are asked during the webinar will be available in a document that will be linked to their webinars page. There's also a materials directory on the FTP side that has Ashley's slides, and this compressed URL will take you there, and I'll go ahead and send that out in the chat pod. So I'd like to introduce Ashley Hentz, who's going to talk to you today about the SRA Run Selector.
All right, thank you very much. Okay, so here we're going to go ahead and get started. What are we going to talk about today? We're going to talk about the structure of the SRA database. We'll go through what the Run Selector actually is, how to effectively use the filters in Run Selector, getting access to the data that you found, and we'll go through a couple examples of the SRA Toolkit commands. So this is a diagram of a standard SRA submission setup. So up at the top we have two studies submitted, and those are directly linked to the bio project, and they will also have an SRP accession number in SRA. Now each one of these different studies has multiple samples linked to it, and these are directly linked to the registered bio samples. They will also have a unique SRS accession. Now you can see there is one sample in the middle called control that is party to both study 1 and study 2. That is perfectly acceptable. Now underneath the study and the samples, we have two more categories, experiment and run. This information is directly stored in SRA. Experiment accessions with the SRX number have all of the metadata provided by the researcher contained within it. And underneath that, we have the SRR numbers, which is our run accessions. The run accessions are where the actual data files for the sequence data are stored. And you can see there are, in some of these samples, one experiment linked to multiple runs, and some samples have multiple experiments and multiple runs. So there are different ways to set up SRA to fit your research project. Okay, what is Run Selector? This is a table view of SRA data available from your search terms, and we'll go through a demonstration of this after I've laid out the basics. This allows for more precise filtering of the SRA metadata. It collates the common columns of data between the projects. With a couple exceptions, there does have to be a 10% similarity between the data columns, and fields with large number of characters will also not be included because it makes the table harder to read. There is a limit of 20,000 run accessions that can be viewed in the Run Selector. And finally, after you've filtered down the data to where you want it, you can export the data accessions and the metadata as a spreadsheet. All right, so let's go ahead and jump into the demo. Okay, so we're going to start from the main NCBI page, and I have SRA database selected on the left-hand side. And this is where you can enter in any search term that you're interested in. But be sure your search terms are relatively specific, so you get below that 20,000 run accession limit. So I'm interested in ancient DNA. I'll click enter. Okay, and this will take me to all of the SRA experiment accessions that we have for those search terms. And over on the left-hand side, you can see there's an access area. I want to point out that you can use the Run Selector on dbGaP data. In this case, we are only going to look at public data, however. So I'm going to go ahead and click that. And you see there is a box right at the top that asks me if I want to send this data to the Run Selector, which of course I do. So we'll go ahead and click that. And after it caches all of the data, you'll see a view like this. You'll notice on the left-hand side there is a box labeled as facets. This is all of the data column headers that you can use to filter the table. You'll also notice that the center is automatically selected. This is just sort of default when you click on Run Selector. If you don't really care which center the data came from, you can click on the little red minus button and it will make it go away. So now I can filter the data the way I'm interested in. And we're going to start with organism. So as you can see, we have lots of different data for multiple different organisms. I'm specifically interested in human data, so I'm going to go ahead and type in that genus. And when I do, there are two different species for the genus Homo. We have Homo sapiens, of course, and then Homo heidelbergensis. I'm only interested in Homo sapiens, so we'll click on that one. And you'll notice the table automatically updates. I had over 2,000 runs, and now I'm down to 486. The next category I'm interested in looking at is the assay type. So we'll click on that one. And again, it also identifies how many runs are labeled as each of these categories. I'm specifically interested in whole genome sequencing, so we'll click on that category. And again, my table will update. I'll make a couple more selections here. I'm going to select a particular library. So we have hybrid selection, unspecified, and then random. I'm specifically interested in random. And finally, I want a specific spot length. So let's go ahead and type in 60. I want them at least 60. So once I type that in, you can see there's only 3 categories that fit that criteria, and I can select all 3 of those. Each time I do, it will update. Okay, so now I have 131 runs that fit the criteria that I'm interested in. Now you notice not all of the accessions over in the table start with that SRR number, or letters, I'm sorry, that we talked about. These actually begin with an ERR. That means it's coming from our European database, our counterparts in Europe. The SRA database mirrors with European and Japanese databases for next-generation sequencing data. So when you search our database, you're not only looking at SRA data, but you're also looking at data from those 2 organizations. So you're getting 3 databases. And I can also sort each one of these columns. So if I click on it, it's going to sort by the letters and numbers. So forward and then backwards, then I can see all of those SRR accessions. In this case, there aren't very many, just simply because ancient DNA is more prevalent in Europe for doing research with. That's why you're seeing a lot more ERR accessions. Now you'll notice the first 4 columns are blue hyperlinks. And I can click on one of these, and it will send me directly to the information for that particular run, study, bioproject, or sample. This one looks interesting just because it says ancient DNA of a metagenome. So let's see what that's all about. I'm going to right-click and open that in a new tab. So this takes me to all of the run data, and I can see how many spots it has, the size, the GC content, and down at the bottom, I can see the title for this project was specifically From a Tooth for a Victim of the Black Death. That's kind of cool. Okay, let's just scroll out here so I can show you a couple more things. You can see for some of these categories there are runs that are blank. The researchers chose not to enter information for that particular category. So keep that in mind while you're filtering these data sets and looking through them. This data is provided to us from research community, so not all of the columns of data will be filled in. In this case, we're doing pretty good. There are some missing collection dates. But let's say all of this data looks good. I want to export it. So what I'm going to do is click the green plus button. It will select everything in that spreadsheet. But maybe I'm not interested in that Black Death victim, so I can unselect that particular run. And so you can see under our selected column here, row, I have 130 runs selected, and I'm going to download that information. And let's save it under download so I can find it. Okay, now I want to open that in Excel. Go to open this PC. We'll get to downloads eventually. Now here I have to make sure I select all files, because when you select to download the run table information, it will download as a tab delimited text file. So you have to make sure you select all files, and we'll open that. Now I have to choose that it is delimited, so we'll click next, and it is a tab delimited file. Again, next, and finish. Make these columns a little bit wider. Okay. So now you can see here's all of that same information that was in the Run Selector is now in my Excel spreadsheet, and I can save it, make notes, go back to it later. The other option for downloading this information is just to download a list of the run accessions. This is the most important accession number that you'll get from this table because, as previously stated, the actual data files are resting under the run accession numbers. So we'll download our accession list. Save that. All right, I'm going to open this in Notepad++. Okay, and here are all my run accessions, all 130. All right, let's jump back into our slideshow here. Okay, so now you have all of this metadata information, all of these run accessions. How do you get access to the actual data files? You're going to use SRA Toolkit. This is a command line application that works on all current 64-bit systems, Windows, Mac, and we have 2 versions for Linux. You can download the data in multiple data formats, the most common being FASTQ, but for any aligned data you can also download BAM files, and then we also have SFF files, among others. You can also limit the amount of data that you download. Maybe you're only interested in chromosome 1, maybe you're only interested in the back half of chromosome 16. You can enter in those sections when downloading the data, and that way you don't have to obtain the entire large data file. Another thing the SRA Toolkit does is blast. So you can blast data from SRA, including barcodes or aligned slices, and that's just the beginning. Okay, here's a couple examples of commands that you would use in the SRA Toolkit to get access to the data. So for a standard FASTQ download, you would use FASTQ dump and the SRR number. This function also includes options for splitting files if it's paired data, and also only downloading a section of the file, as previously mentioned. If you want to download a BAM file, make sure again you have to filter by aligned data specifically. You also need to have SAM tools installed on your computer because they work together. So you're going to use SAM dump and the SRR number to get your SAM file, and then take it directly into SAM tools to convert it into your BAM file. And then finally, an example blast command. So it's blast n_VDB. And in this case, you can see there are 3 different run accessions that we're querying against. And the other thing to point out is this nt.test file. This is the actual sequence you are searching for. So here's the nucleotide sequence to see if it's in any of these run accessions. And then .out will give you the results. Okay. Here are the links for all of the information that we talked about today, including some additional information about the SRA database. And with that, I'm happy to take questions.
Just to remind you while we're open like this, just if you have any questions about any of the resources you heard about today or any NCBI resources in general, you can write to our info address. That's info@ncbi.nlm.nih.gov. That's the same domain name as our webpage. If you have questions about the webinar program or webinars in general, technical details about the webinar, please write to the webinars address, webinars@ncbi.nlm.nih.gov. Okay, I don't see any questions that we can answer right now. So thank you very much for attending the webinar today. And that's all for today. We'll see you next week on the 30th for a webinar about the NCBI hackathons. Thanks a lot, and thanks for coming. Thanks, Ashley.